Hello, Bio30. This lesson is on, as the title says, the work of Gregor Mendel. Gregor Mendel is considered to be the father of genetics. He did his work in the middle part of the 19th century. In fact, he did his work at about the same time that another famous biologist, the most famous of all, Charles Darwin, was publishing his work on the origin of species. And of course, whereas Charles Darwin is the father of evolutionary thought, Gregor Mendel is the father of genetics. No doubt that Gregor Mendel would have known about the work of Charles Darwin. Charles Darwin was fairly famous because of his publication on the origin of species, but probably pretty much guaranteed that, unfortunately, Charles Darwin knew nothing at all about the work of Gregor Mendel. In fact, very few people knew about the work of Gregor Mendel, nor did they care about the work of Gregor Mendel, at least while he was alive. So who was Gregor Mendel? Well, he was someone that did have a background in mathematics. He did go to the University of Vienna, study mathematics, so he did have a science background. He was a monk that lived in a monastery in Austria, and there he was a teacher. He was also interested in science. He was also interested in performing some experiments. And like a number of people that were in monasteries or religious people at the time, they had a little bit of spare time, unlike most of the general population, I suppose, time to perform these different investigations. And that's exactly what he did. Initially, what he did is he went to the bishop in the monastery and he said, "Hey, I'd like to take a look at mice and study the reproduction in mice." And the bishop said, "No, no chance of that, because we can't be taking a look at and studying sex in a religious monastery." So then he said, "What about bees?" And the answer to that was no as well. So eventually, as we'll see, what he did study were garden peas. And in fact, he studied sex that was taking place in garden peas. Prior to the time of Gregor Mendel, most people thought in or believed in what is referred to as blending inheritance. So if you take one parent with one particular trait, another parent with a different and distinct trait, then what you would get in the offspring is a blending intermediate between the two of them. In fact, what Gregor Mendel showed, at least with his garden peas, is this is absolutely not going to be the case. So what he did propose was that there were packets or discrete units that were passed on from the parents to the offspring, and this is the basis behind his fundamental laws of heredity that he did develop. He didn't know about chromosomes. No one knew about chromosomes. No one has seen chromosomes. No one knew about DNA or what chromosomes were made up of, or genes or anything else like that. So he developed these laws without any of the basic understanding in terms of what the basic units of heredity actually are. So he did a fairly extensive study that did last over a period of 8 years, and somewhere on the order of 30,000 peas that he did grow and observe, various different crosses that he did perform. We have at the upper right-hand side here all of the different easily identifiable and observable traits that he did take a look at. Very, very fortunate in his choosing of this particular species or strain of garden pea that he did, because in all of these cases it was either one or the other in terms of what they looked like. So it was purple or white, it was axial or terminal in terms of the flower position. The seed color was either yellow or green. The seed shape was round or wrinkled. The pod shape inflated or constricted. The pod color green or yellow. And the height either tall or short. So very easy to identify these. And that's exactly what he did. So after an 8-year period, he did publish his findings, but it was in a rather obscure publication that no one really read. It was in a foreign language that most scientists were unable to read. And because of this, very unfortunately, it sat for multiple decades, like about 4 or 5 decades, until it was rediscovered. And it wasn't rediscovered until, in fact, a couple of decades after the death of Gregor Mendel. So why did he take a look at the garden pea? Again, he was really, really fortunate. Sometimes in science, luck is involved, and he was definitely lucky for choosing this as his study sample that he did take a look at. So garden peas, available, cheap for him. Easily identifiable characteristics, as already mentioned. Self and cross fertilization. Plants are kind of unique. On the flowers, they have the equivalent of both the male and the female reproductive organs, producing both the egg and the sperm in one single plant. So what that means is that we can have one plant, the sperm from one plant, fertilizing the egg from one plant, and that is referred to as self-fertilization. If we take the sperm contained in the pollen from one plant and the egg from the other one, that is referred to as cross-fertilization. And the last one that I have here, relatively short generation, so he could grow a couple of generations of these plants every single year. So Gregor Mendel, again, did have a background in science and in particular a background in mathematics, no doubt about it. People had been growing garden peas for, well, probably thousands of years, and both self and cross-fertilizing garden peas for at least hundreds of years. So people would have been able to perform these crosses that Gregor Mendel did, but the one thing that he did differently, having a mathematical background, is he counted. He counted what he did get for his results, and he made some statistical analyses. And from this, he was able to come up with, well, the foundations of how traits are passed on from one generation to the next by making these numerical comparisons. Before Gregor Mendel did any of his crosses, though, the first thing he did is he developed what are called true breeding or pure lines. So he would have plants that would produce purple flowers and only purple flowers generation after generation after generation, whether they were self or cross-fertilized. He had his cluster of purple flowered pea plants. And he would have the same thing for the white and the axial flower position, the terminal flower position, and all of these different traits that he did take a look at. So that is what he used as his parental generation, and then he went ahead and performed a whole bunch of different crosses. What he got in the first generation, so the offspring of the parents, he called that the filial 1 generation or the F1 generation. When he took that generation, the F1, and crossed them, he called them the F2 generation, and so on and so on. So this is what he did over an 8-year period, and we'll take a look at a couple of his crosses that he actually did here. So this first one, this is a cross-pollination. So again, the pollen with the sperm is coming from one flower, and the egg is coming from the other one. So what he was crossing was parents that have purple flowers with parent plants that have the white flowers. So the way that we set this up, capital P is what we use to represent the parental generation. In the first row, we write down what those parents look like. So one of the parents purple, this big multiplication sign just means that that is what we are crossing it with, and it is the one that is white. And we can just see a picture showing this at the right-hand side as well. When we see this arrow going down, what it means is that this is what they are going to produce when you do cross these two parents. This is what they are going to generate in the next generation, which is the F1 generation. So what Gregor Mendel did find is that all of them, 100%, not most, not some, but all of them were purple in the F1 generation. So then the question is, well, what happened to the white coloration from one of the parents that we did find in the parental generation? So Gregor Mendel did exactly the same thing with all of those other 6 traits that I showed you, and what he found was, well, exactly the same thing. One of the traits, it's as if it disappeared in the F1 generation. Gregor Mendel also did what is referred to as the reverse cross. This is kind of significant and kind of important. So if Gregor Mendel in the first cross took the pollen and the sperm from the purple flowered pea plant, remember this symbol here is for the male, so take the sperm from the purple flowered pea plant and the egg from the white flowered pea plant. He did the first cross, and this is what he got. All of them were purple. The reverse cross means that, well, now he's just going to flip it around, and now he's going to take the egg from the purple flowered pea plant and the pollen from the white flowered pea plant, and it just so happens that he got exactly the same thing. So this is kind of important because if he hadn't done that, there could have been some misconceptions. If he had not done this reverse cross, then one of the misconceptions or one of the mistakes might have been to have said that, well, this trait comes from the male. It's the male that passes the trait on to the next generation. So by doing the reverse cross, what he did show is that no, it actually has nothing to do with gender. There is something else that is at play here. We'll see this with another cross as well. When we do take the F1 generation and cross them, that trait that disappeared in the F1 generation, in other words, the white flower color with the pea plants, disappeared in the F1 generation, but somehow it reappeared in the F2 generation. Not only that, but it was always 1/4, 25% of the offspring in the F2 generation that had the white coloration, the one that disappeared in the F1 generation. Let's take a look at another cross here, and this one does show both the F1 and the F2 generation. So this one is for the seed shape, and again, we can see a picture of the seeds on the right-hand side. So round and wrinkled, cross them, and what he found was, again, one of the traits, the wrinkle, that disappeared in the F1 generation. All of them were round, but that trait that disappeared in the F1 generation reappears in the F2 generation. So if we do take a look at these two numbers here and calculate the ratio, it is amazingly close to this 3-to-1 ratio. Again, what that means is that 3/4 of them are round and 1/4 of the offspring in the F2 generation were going to be the wrinkled. A few terms, important terms for you to understand before we do go on. These are Gregor Mendel's terms, dominant and recessive. So when we talk about a dominant trait, first of all, what it is not is the one that is more common. Dominance has nothing to do with how common it is. Dominance has a different definition when we're talking about genetics. So in terms of what we saw, the dominant trait is the one that was observed in the F1 generation. So the two crosses that we took a look at, that would be the purple flower color and it would be the round seed shape. This determines what is really seen or expressed. So what did we see in the F1 generation? We saw the purple coloration, we saw the round seed shape, so those are the ones that are dominant. We're going to use some letters to represent these different traits. And what we use if it's dominant is always a capital letter. And what we choose is the first letter for that particular trait. So if we take a look at what I have at the bottom here, round seeds we saw in the F1 generation, we didn't see the wrinkled, which means round is going to be dominant. What do? You take the first letter of that dominant trait, R for round, you capitalize it, and that capital letter R is now the letter that we are going to use to represent the set of instructions that are for producing the round seeds. What it also does is, when it's present along with the other version, which is the recessive version, it's going to mask it. So it's going to hide it as if it isn't really there. It might be there, but it's not seen, it's not expressed in terms of what the plant in this case looks like. So the other one, yes, is recessive. So that is the one that was not seen in the F1 generation, but reappears in 1/4 of the F2. These genes are the ones that are being overshadowed by the dominant version of it. In this case, we use a lowercase letter, as we can see, but it's not a lowercase w. It is the same letter that we used for the dominant trait, which was the R, only now we're going to make it lowercase. So capital R is for round, lowercase r is for wrinkled. So these are Gregor Mendel's terms, but there are a few other terms you need to be aware of as well. We've sort of been talking about genes. We'll talk more about genes later, but for now, a gene is a set of instructions made up of DNA that you find on a specific location or at a specific location on a given chromosome. And for our purposes, at least right now, it is responsible for the production of a single protein. So what are some of the genes for? A gene for seed shape, a gene for pod shape, a gene for, as I have here in this example, the pea color. An allele is just a different version of the gene. So there can be various different versions. It can be 2 or more different versions. So green, yellow, those are 2 of them that we do see with Gregor Mendel's garden peas, maybe with some other peas. There are some other versions as well, perhaps the orange, but alleles are just different versions of instructions for the same gene. Very important word, genotype is the actual letters that we're going to be using. It is the genetic makeup that an individual has. You cannot see an individual's genotype just by looking at them, but the different possible genotypes that we could have in terms of letter combinations when we're talking about adults. Remember, adults have 2 copies of each chromosome. They are diploid. So when you're representing the genetic makeup for an adult, you do have to have 2 letters. So if we're talking about the R's, it can be 2 capital R's, 2 lowercase r's, or 1 of each. Those are the 3 different possible genotypes. Phenotype is what you typically can see and sometimes what you can directly see. So it is what it would actually look like. So it is a tall plant or it is a short plant. Those would be different phenotypes. The last 2 here, homozygous in terms of the genotype. Homo means the same, it means the letters are the same. So we can either have what is called homozygous dominant, both of the letters are capitalized, homozygous dominant. If they are both lowercase letters, then they would be also homozygous, only they would be homozygous recessive. 2 of the same letter, but in this case for the recessive allele. The last one is heterozygous or heterozygote, and that just means that instead of 2 letters being the same, there is 1 of each, and the example here would then be a capital R and a lowercase r. So let's go back to the original example here, and let's assign some letters that we would be using for the original cross that we talked about with the purple and the white flowers. So what did we see in the F1 generation? We saw purple. That tells you what is dominant. How do you assign letters? Take the first letter of the dominant trait, capitalize it. That's what we're going to use for the purple color. Lowercase p, not a W, is going to be for white. Parents are diploid, 2 copies of each letter. So if these are the original pure breeding plants that Gregor Mendel did develop, pure breeding is the same as homozygous. So this purple one would be homozygous. There has to be a capital P to be purple, and if it's homozygous, the other letter would be a capital P as well. White, the only way to get white, 2 lowercase p's. So when reproduction does take place, remember through the process of meiosis, each one of these plants is going to produce gametes, but the gametes are only going to be haploid. Gametes are only going to have 1 of these letters, but each parent, they only have 1 possible letter to contribute. For the first parent, that's going to be a capital P, and for the second parent, they can only contribute a lowercase p. So if we take a look at these plants, they are purple, just like the one in the original parent population that was purple as well. They look the same, but notice that the genotype can be different. Why is the genotype different? Well, both of them have the dominant allele, the capital P, and as long as they have that, they have the instructions for the purple coloration. Different genotypes generating the same phenotype. Let's do the same with this one here. We already know what the letters are. This was in the example. So the capital R was for round, the lowercase r was for wrinkled. These were the original pure breeding parents then, big, big, and little, little, for the round and wrinkled respectively. What we get in the next generation is the round seeded parent can only contribute gametes that have a capital R. The wrinkled seeded parents can only contribute gametes that have the lowercase r. Just as a reminder, once again, gametes are haploid, parents are diploid. Parents diploid, gametes haploid. 2 letters for the parents, 1 letter for the gametes. So what we then get, the combination of these in the F1 generation, is 1 of each, a big R and a little r. So now we're going to take these and they're going to become the parents for the next generation. So now what we would have to generate these different phenotypes is we would have this parent and this parent here. So what are the different possibilities? Well, now what we can get is we can have this gamete, the capital R, ending up combining with this one here. So we would expect some of the plants in the offspring, the F2 generation, to be homozygous dominant. We could also get the big R with the little r from the second parent. That's the second possibility. We could get the little r from the first parent and the big R from the second parent. For a given gene, you write the capital letter first. So these two, in fact, end up being the same genotype, both heterozygous. And the last possibility that we have is a lowercase r from the first and from the second parent. Those are our four different possible genotypes that we now have when both of the parents are heterozygous. This is what we would expect for the offspring. So now if we take a look at these, out of the four different possibilities, three of them actually look the same because they have a capital R. And that's where we end up with this one here, the 3/4 that are round. The only one, the 1 out of 4, this one here, is going to be for the wrinkled. Just to remind you again of a little bit of the language that we're using as well. So this first parent that we had up here, they are homozygous for the capital R's. And again, what we call this is homozygous dominant. So we're crossing a homozygous dominant round-seeded pea plant with the other one, two lowercase r's. So that was the homozygous recessive parent. In the F1 generation, what we're getting are plants that have one of each, and this one is called the heterozygote. And we'll just take a look at one more final example here of another cross that Gregor Mendel would have done. And this one is now taking a look at the seed color. This one does have the letters that are already indicated for us. And if we simply take a look at the letters, we're using a capital Y. That tells us that the yellow is going to be dominant. So again, this yellow seeded one, that's going to be the homozygous dominant. The other pure breeding parent is green, homozygous recessive. F1 generation, they get a capital Y from parent number 1, a lowercase y from parent number 2. What we see though is that the phenotype for the heterozygote is going to be the same as the parent that is homozygous dominant because they both have a capital Y. And we continue on to the F2 generation. Very important that you recognize this particular genotype. This is the genotype that we get when we are crossing two parents that are going to be heterozygous for that gene. So again, this is for the F2 generation, which means the parents that we're crossing are both heterozygous. That gives us the 3-to-1 ratio, but it's also important for you to understand and indicate that the 3 is for the yellow and the 3 is going to be for the one that is the dominant trait. The 1, make sure you specify that it is for green, and the 1 is going to be for the one that is recessive.